Saliva also solubilizes dry foods so that they can be tasted by the tongue. It contains the major taste buds in the oral cavity, which contains the cells that detect molecules from the food. These cells form clusters of 50 to 100 columnar cells resembling a garlic bulb, and serve as checkpoints for quality control, distinguishing between pleasant and unpleasant tastes. Most poisons, for example, taste bitter, while off food tastes sour, and energy-rich foods taste sweet or umami. Taste buds are classified into 3 different types. About half of the total number of cells in a taste bud are type 1 cells, which have glia-like functions, expressing enzymes and transporters that eliminate extracellular neurotransmitters. Approximately 1/3 of the cells in a taste bud are type 2 cells, which are larger in diameter and function as chemosensory receptors for sugars, amino acids, ions, and bitter stimuli. Type 2 cells respond to one taste quality, such as sweet or bitter, but not both. Type 3 taste cells are the least numerous and represent 2 to 20% of the cells in a taste bud. These cells sense sour tastes and also respond secondarily to other taste stimuli via cell-to-cell paracrine communication within the taste bud. When you take a bite of those spicy beef tacos, the amino acids in the meat bind with receptors on the cell membrane of taste bud cells. This causes the intracellular part of the receptor to change shape, attracting and binding with G proteins, inducing G proteins to exchange a GDP ligand for a GTP molecule, which causes the separation of their alpha, beta, and gamma subunits. The alpha subunit of the G protein then activates phospholipase C proteins, which contain an enzyme that takes phosphatidylinositol bisphosphate molecules from the membrane and hydrolyzes their inositol part, releasing dozens of inositol molecules into the cytoplasm. These inositol molecules serve as ligands to inositol-dependent calcium channels in the endoplasmic reticulum, inducing them to open and release calcium ions into the cytoplasm. Calcium ions serve as a ligand to sodium channels in cell membranes, inducing the release of sodium into the extracellular space, causing a reversal in membrane polarity, and initiating an action potential that spreads through the whole cell membrane. The change in membrane voltage causes non-selective ion channels to open, releasing ATP as a neurotransmitter at a synapse with an efferent neuron. ATP serves as a ligand that opens ion channels called P2X, causing depolarization of the membrane in the taste nerve and initiating action potentials that travel down the efferent neuron of the taste nerve. The taste signal is transmitted by the glossopharyngeal nerve to neurons located in the nucleus of the solitary tract.